May I introduce the next speaker, Rudolf Leube. So this is another biological topic, and he will talk not about single cells, but about adhesion in epithelial cells and migration and implication for migration. That's adhesion between individual cells, and that will be about adhesion between the cells and the surface from a more biological view. Rudolf.
All right. Having heard all these wonderful talks, I feel I'm on really, really thin ice here, because what I will be doing is mostly giving you some very rudimentary and basic concepts, ideas about cell-cell adhesion, and not so much talk about results from my laboratory. But since I will be here for 6 months, it will be ample opportunity to do that later on another occasion. Now, if you think about thank you very much. If you think about cell migration, that's probably what we will be dealing with mostly during probably the entire period. Single cells walking across a field, and we've heard quite a lot about that these cells, in order to move, they have to interact in a sort of a transient fashion with the extracellular matrix to actually propel themselves forward, if it's not a liquid, but for adherent cells. And that's what I'll be talking about adherent cells, about epithelial cells. And if you actually look at this process of adhesion, deadhesion can be imaged by looking at new formation of junctions, dissolution of junctions in polarized cells, which has to be somehow coordinated with the intercellular cytoskeleton. Of course, since we're working on keratins, you see here the keratin intermediate filament system, how it sort of comes along with this cell that's moving here to the upper left corner. But this is certainly only part of the story, because if you actually think about a wounding process or many other situations, we're not talking about single cells walking across an empty surface, but we're actually talking about monolayers. And this is one such scratch wounding assays, which many laboratories do. You take a little pipette tip or a needle, you scratch it across a monolayer, and this is this sort of dark gray contour lines that you see here, and then the cells will actually move towards each other and meet each other in the middle, and this is sort of thought is an example of a wounding situation. And you can actually use this type of assay and look at various parameters. Of course, you can do imaging, like in this instance. Again, you look at the intermediate filament system, and you see that these cells don't migrate as individual cells, but they migrate as a sheet. So when you talk about cell-cell or epithelial cell migration, we're not only talking about interactions from the cell to the extracellular matrix, but also about interactions from between individual cells, as shown in this instance here. Now, there are various ways of actually examining this kind of behavior, and I really like this type of assay where you don't use this needle, which is sort of more or less perfect, but instead you use sort of a stencil, and use this stencil to culture your cells in confinement. So what happens then is that you have a very dense, coherent colony where all the cells are connected actually by cell-cell adhesion sites. You remove the stencil, and then you can actually see how the cells migrate out. So here at the bottom you have various stripes of different size and different gap length, and you see within like a day these gaps are being filled. And the gaps are not filled by individual cells sort of probing and running along, but by actually collective sheet migration, and that's what we're talking about in this situation. And this collective sheet migration or cell migration relies on interactions through individual cells, which are actually transmitted two neighboring cells, and this is a displacement field here, which is done by image analysis, and you see that there are sort of groups of cells that are put together and probably are tucked together in this situation, indicative that there are really long-range interactions which not only go from one cell to another, but probably extend over 10 or even more cells within such a collective, in such a cell sheath. Yeah.
Is that PID or something else?
Yes, it's PID. PID is PDMS, right?
PIV.
PIV, yes, it's PIV, yeah. Particle image velocimetry, yes.
Is there cell division going on here?
That is a very good question, because clearly for definitely for the scratch wounding assay, probably what many people measure is actually not cell migration, but just cell proliferation, which I think, at least in this situation, is less relevant than in the scratch wounding assay. But yes, it's something you have to really consider, because the cells have a division rate of 24 hours or something, and you can almost close the gap just by proliferative pressure from behind. Very true. Now, I really love this experiment. It was published by the Ledoux group last year, and they used a very simple assay here. So they have the cells grown in confinement here by just putting a PDMS block on top of it, and then they lift this block, and they have a repulsive surface just around this red marked area, and within this repulsive area they have stripes of fibronectin, so to say, little sort of passageways that the cells can use. And when you then take off the PDMS block here, you see that the cells here move along these lines here. And that's probably also something you will find in vivo in the situation, because you have a denuded surface, and maybe the extracellular matrix is not distributed homogeneously anymore, but in certain areas only present. And if you then watch what's happening, is that actually you have these protrusions here. There are certain cells that really use these tracks to migrate and actually pull other cells along. So the distances between these stripes can be several cell diameters. So it can be multiple cells in between, and they sort of get dragged along about this repulsive, non-adherent surface while these cells move up here. Yes?
This means that in between the cells are flying over the substrate?
That's what you see here. This is this picture, exactly. They are flying. It's a good way to so you look from up here, you see here these red stripes, which are little, ridges, so to say, and the cells, these are the cells that migrate here, walk along these ridges, and in between you have the cell sheath, which is pulled along in the air, basically, or in the liquid, of course, in this instance, and get pulled along. And they also provide an animation of the whole situation, and you can really nicely see how these cells get dragged along. So there's tension in this whole cell sheath. And this works in the absence of the extracellular matrix in between. This whole sheath is being pulled up here. Another sort of situation that shows you that this sheath migration may actually be quite important. Yes, please?
Backtrack a little bit.
Yes,
it looks like the bridge is not formed by one stream of the cell jumping over the gap.
No, it's multiple cells in between. This can be 4 or 5 cells in between here.
I know, but still, it looks like it's not a bunch of cells making the connection in midair, but the detachment from the video below, it looks like there's a de-wetting, so to speak. The formation of a dry spot. Is that how the bridge was formed? If you run the video again, you will see the appearance of the dry spot.
Well, there's something else.
In the hole.
You mean the hole here?
Yeah.
That's actually caused by cell division in this case, in this particular instance here. And the explanation they bring in this paper is that through cell division you actually have a loss of local adhesion, but the whole sheath is still together here. It's completely together. You mean this here, right?
That's right.
Yeah, but this is due to local cell division.
Nothing to do with the bridge.
And they can keep going forever?
Well, even those people had to go home, have family, wives, friends, and then stop their experiment. But yes.
Yeah, okay.
But then actually that hole is interesting because it's like doing a laser ablation experiment. It's telling you that the tissue's under a lot of tension.
Exactly.
Because the moment you reduce the coherent adhesion, the tissue falls apart.
That's why they actually show it in here. That was the reason why they actually used this particular example.
Have you studied which has to be the minimum or maximum distance between the pillars?
They did that, I don't remember this exactly, I think up to 10 cells that's possible.
10 cells.
Yeah, but maybe wrong. But it's quite a number of cells, yeah?
One step. But is there something different at the leading edge, this white front, or?
Yes, there is. And actually you can see it in another setup much better. This is this here. So in this case you have also a confined cell, and then they use the collagen matrix, the collagen matrix, and then you also have collective sheet migration, and look at these cells here, how different they are. So there are leaders, like in our society, and there are followers. So leaders are telling them what to do, and they did a very nice experiment for this one here. They actually, to test the leaders, and actually we've heard about the GTPases, the RUC GTPases. So what they do here is they, so it's all, this was on, let it run again. Let's go from the beginning again. So here you have the sheet migrating to leader cells, then they add an inhibitor of lamellipodia formation. It stops the whole thing, and you can wash it out, and the whole thing goes on again. Yes?
Is the identity of the leaders and followers changeable? Can you take cells from the leaders and put them at the front, and will they become leaders?
It's difficult, but it is probably possible.
Actually,
You can kill the leaders and they stop. Yeah.
I've heard Joachim Spatz speak about
Okay.
that, and he claims they can exchange.
Yeah. Lots of people think they can.
They can.
Actually, I know some experiments. Oh well, but this is the one by Gilmore, right? This is the zebrafish, right?
Oh, that's the next one. Yeah. This is not the zebrafish. This is a culture-assisted NDCK cells, and this is an epithelial cell line.
In the zebrafish, if you cut the head,
Yeah. You
other cell will take over and make the head, make the leader.
well, you saw this from Bernd already this year. This is just to prove the point that actually this sheet migration or collective migration is relevant for in vivo situation. Yeah. All the work from Gilmore here in the zebrafish in the lateral line organ, where you actually see, and the situation is a little bit different there, because they are sort of chemotax toward this gradient, and they have sort of mesenchymal type leader cells, which then transform into the rosette types that you see back here, which is more the epithelial phenotype in this case. And the last example I selected here is sort of suggestive, and I hope we will be able to hear more about this from Peter Liedl or somebody from his group, is about tumor invasion, where you also have sort of the same type of arrangement here, and he's actually spectacular movies where you see a whole group of cells moving in the spaces between the different tissue types. And he's also the one who sort of conceptualized this quite a bit with respect to tumor formation. And if you're talking about epithelial cells, these are the cells that make cancer by definition. Epithelial tumor is a cancer. And if we are talking about epithelial cancer, probably the amoeboid type of movement, which we've heard a lot about, is probably the more atypical one. Although it would be that one when you have an epithelial cell losing its epithelial phenotype and sort of going into a more mesenchymal type of cell, but that would be the individual cells. Probably much more important is that these cells actually invade tissue as clusters in different kind of spatial arrangement, as strands or as sheets, and not as single cells. And even you can even make the argument that probably also for metastasis, not people really haven't
seen that single cells actually go into the long journey, but maybe also little cell clumps, little cell clusters, and actually clinically this is known that tumor form emboli upon metastasis. And what's the message for today is that this is dependent on specific adhesion molecules to the extracellular matrix and also on cell-cell adhesion molecules that correlate with this type of behavior. So two things we need. We need the pulling forces from the leader cells by attachment to the extracellular matrix, and we need the pulling along or dragging along of the followers to anisotropic pulling stresses, and this is mediated through cell-cell adhesion. Okay. This is the major message I wanted to give for today. Think about it. We're not only talking about single cells, but about actually whole tissues or whole collections of cells. And I will now go on a bit more in the nomenclature and bore you a little bit with names and with terms that we use to describe junctions. Yes?
Just in the previous slide, you want to be a little bit careful because it's not that the leading edge cells are pulling on the whole tissue by itself. If they did that, it would take forever for things.
There's not enough force there.
It's the difference between a coal train with a single locomotive and 100 cars behind it and a subway car where each car has its own engine. Mm-hmm. And so it's really a transmission of an inductive signal to move, not just that you're being pulled. Mm-hmm. And so I know you know that, but the way this slide is written, maybe people who haven't read it.
Well, I kind of insinuated it too by my suspended cells is a very, very important and good point. And I really encourage you to actually comment on what I say, because this is more done by reading than by knowing many of the things that I'm talking about.
Comment?
Yes.
But that's only partially true, because if I take an aggregate of cells, it's only the cells at the exterior in contact with the extracellular matrix or the substrate that can actually do work. The ones in the interior are not contributing anything to the forward motion.
We don't know that. They can also attach to the extracellular matrix quite so.
Well, but if you have a cylinder of cells, right, then it's only the ones around the periphery that are actually doing the work of moving the aggregate forward.
But the cell interactions can push on each other. The cells themselves, hopefully they'll push on each other.
If I'm in the interior and I'm pulling on you to try to get ahead, I'm pulling you back, right?
No,
so
but
in the interior, sure they do.
No,
we
but
showed
Hans, in wound healing, if that were the case, the velocity would decay exponentially as you go away from the front.
But they're all in contact with the substrate. Here we're talking about, in the case of metastasis, we're talking about the possibility of a core with some finite thickness moving, and I'm claiming that it's only the ones around the periphery that are doing the work. We can talk about this later.
Sure, okay, no, we're
This is
different.
again the difference between 2D migration and 3D
yes,
migration.
sorry, no.
He's talking about 2D migration, and you're talking about 3D migration. It's full core, right?
If you're playing the game of tug of war, then everyone playing tug of war is contributing, unless they happen to be hanging from the rope and not touching the ground.
Yeah, it's a
I'm sorry, I didn't hear you.
If we're playing tug of war, and we got 10 people on one end and 10 people on the other end, all 20 people are contributing to the tension in the rope, unless there's somebody in the middle who's just kind of hanging on.
Yeah, but then everybody's in contact with the substrate, right?
It's
What
a
I'm
piggyback.
talking
That
about,
is not true.
what I'm talking about is if you have a solid core of cells, it's only those around the periphery in contact with something to which you can transmit the force that are actually doing the work.
All right. So I'll be talking about now the components of the system. And this is again the keratin filaments. And of course, you've all heard this, that if you look at the cell, it's not just a drop of water, but you have the rigid cell cortex is connected to the cytoskeletal filaments, various types that are attached to the more rigid nuclear lamina. And these individual systems in individual cells are actually coupled to specific cell-cell adhesions to each other and to cell extracellular matrix adhesions to the extracellular matrix, which in a simple one-layered epithelium would be just the basement membrane. And the basic idea is for the resilience of such an epithelial tissue, depicted a long time already in the standard cell biology test book. You pull on the system, and it has sort of an intrinsic capacity to withstand this sort of tension through these cell-cell adhesions in the cytoskeletal filaments. And this is the experiment been done a few years ago by Doug Fudge. And you see this when he uses an elastic membrane on which he cultures the cells. He can actually stretch these cells by quite a bit and release the whole thing. And at least for the keratins, you just heard this, they're the elastic component of the cytoskeleton. They don't break, they just recoil and go back in their original position for the most part. So how about the cell-cell adhesions in epithelial cells? And now I would go a step back to actually a tissue situation. Yes?
Sorry, just I could not read the reference here.
This is Berriot et al. 2012 in +1. Okay. So we're talking about how does it look in a static tissue, and here in a simple epithelial, one-layered epithelium, the prototype would be the intestinal enterocyte, where you have the apical site, the basolateral site here to the basement membrane. And what's characteristic for these types of cells, that they have a very highly ordered arrangement of cell context. Always starting at the top, the most apical part, just below the microvillar brush border, are the tight junctions, also referred as the sonular occludens, which form a tight barrier between cells and also within the plasma membrane, so they are responsible for polarity and for any kind of transport process in an epithelial. So this would be here and here. Below it, you have a subcortical sort of ring-shaped arrangement of actin filaments that actually go along the plasma membrane that form the sonular is a girdle adherens, or the adherens junctions, which serves as an anchorage site for the actin filaments, which are here just looked at en face. And then below you have often several, on the basolateral side, desmosomes or maculae adherentes, which anchor the keratin intermediate filament system. And then, not shown in the electron micrograph, but here depicted in this scheme, you have the gap junctions or nexus, which are responsible for electrical and metabolic coupling of adjacent cells. Just for the nomenclature, the adherens junctions and the desmosomes are both types referred to as adhering junctions as anchorage sites of cytoskeletal components. Now, we are lacking a lot more in our knowledge, actually, in multiple stratified epithelia, like the epidermis, where you have not a single epithelial cell layer, but you have a basal cell layer and various other cell layers on top of it. So for spinal cell layer, granular cell layer. And it's still fairly recent, only like 10, 12 years ago, that we learned that we also have, in addition to the barrier function of the dead cell layer, the cornified layer, these cells are also attached to each other by tight junctions, which are part of the epidermal barrier. And then throughout the living cell layers of epidermis, we find adherens junctions and desmosomes. And just with respect to the abundance, up to 80% of the surface of a keratinocyte can be covered by desmosomes. And if you look in the interior of the desmosomes, you have the keratins, which actually can be as abundant as actin in muscle cells. So actually keratins in this particular cell are extremely abundant. And then you have specialized junctions at the basal cell layer, where at least in skin, the hemidesmosome, we'll say a little bit more about this, mediates the attachment. And we're only now thinking that maybe some sort of a correlate to the focal adhesion may also exist in epidermis, but we really don't know what the structural correlate is for this at this point. And if we think about the contribution of these cell-cell adhesion sites and the cytoskeleton, in particular the keratin desmosomal scaffold is important. for actually maintaining the beauty of our skin, but also the histological appearance of it. And this just shows you one such desmosomal adhesion site, which in various diseases, in this case actually a deadly autoimmune disease, can lose adhesion, and then the two cells fall apart, either here in the electron micrograph or here in the histological section, which has very bad consequences for the patients, because this dissociation will result in blister formation and subsequently also in infections which are very bad for the patients. So this is dissociation of cells to actually autoantibody mediated. The kind of disease groups are the pemphigus diseases, which come in various flavors. And similar, just showing that this is all one system. If there are, in this case, a large group of genetic diseases affecting the keratin system, and Rainer just said this, you will find weakening of the cytoplasm of the cells, and then upon mechanical trauma, the cells will rupture. You will have cytolysis and again blister formation, and actually exactly in the same cell layer where this particular keratin is localized, as you would expect. So you have blisters at the bottom or at the top. There are various types of blistering diseases. Now, if you think about it in very systematic terms, what does it take to make a junction, especially an adhering junction? You need adhesion molecules. Oh, this is difficult to read. And there are different types of adhesion molecules, those that are dependent on calcium, that are caterins, those that carry structural similarities to the immunoglobulins, so they are immunoglobulin-like, or those that are membrane proteins going to the membrane 4 times. That's why we refer to them as tetraspannins, and also the integrins, which we've already heard something about. So these adhesion receptors are transmembrane molecules, are attached to cytoplasmic linker molecules, the catenins, also vinculin, or the plaquin, which then allow the attachment of cytoskeletal filaments, which at least in human cells is restricted to actin filaments and intermediate filaments. The connection to microtubules is much less characterized, and it's not much known actually about how they actually interact with these junctions. And if you now look at the specific molecular components, and I apologize, I really like to go through this because I'm a medical person, and in the end, if you know the components, you also know a lot about diseases, you know a lot about disturbances of the system, and you need to know the nomenclature. So among the adherence junctions, we have the desmosome and the, without the A, adherence junction. This one couples to the intermediate filaments, this to the actin system. They differ a little bit in the ultrastructure. The desmosome has the 10 nanometer filaments associated with it, and is characterized by a midline in the middle here. And each, and then these junctions also carry electron-dense material, the so-called plaque material, or desmosomal plaque, or adherence plaque, which is less pronounced in the adherence junctions here. Here, no midline, somewhat fuzzy material, which is the thinner actin cytoskeleton attached to it. So we have transmembrane proteins, which are all of the calcium-sensitive type, the desmoglians and desmocolins and desmosomes, and the so-called classical caterins in the adherence junctions. We have the linker molecules, which in the desmosome are grouped to the plaquin, desmoplaquin, and to the catenin type placoglobin, and placophilin is sort of related to it. Whereas in the adherence junctions, we have various catenins, and I actually come back to the P120 catenin and alpha-catenin later on, and also vinculin. And then, of course, we have, in this case, the intermediate filaments, and here the actin filaments. So the assembly of these molecules here defines the specificity for either the actin filament or the intermediate filament system. And these are the sort of the core component. There are probably additional components, like in this case for the adherence junctions. We now know this doesn't work alone, but is working in cooperation with actually transmembrane receptors of the immunoglobulin type, the nectins, which then attach to the linker aphadin, which is in itself again an actin binding protein. to show how this system works, I just present you here some examples from our laboratory. Here you have a confluent monolayer again of the keratin cytoskeleton, and if you zoom in at the cell border, and here in red label the desmosomes, you sort of see how the filaments seem to end there, but at least for the intermediate filaments, they don't end there, they sort of loop through it, which is quite in contrast to the actin filament system, which has a plus and minus end. The intermediate filaments don't have that, so they don't end there, they just loop through the whole system. And you see that apparently these cell junctions have some sort of coordinating activity on the cytoskeleton of either side, here on top and here on the bottom, as shown in this little example. How do these cations work? The cations need calcium for their function, and they are characterized by 5 repeats, or 4 depending on which school you adhere to, cation-like domains, and these domains, or in the hinge regions between these domains, they are calcium binding sites. So what's happening here, we can see it here better, between this, in this case 5 domains, you have these calcium molecules bound to the extracellular domain, and this binding of calcium actually results that the whole extracellular domain stiffens, becomes it becomes stiffer and can elongate, and can then interact with another cation on a neighboring cell, and they form this sort of interaction in their first extracellular domain, which they sort of interlink with each other and form this rather stable cell adhesion bridges in this case. And if you then remove the calcium, then the stiffening is lost, the molecule becomes sort of wobbly, and the consequence will be that the adhesion sites or the cations are then taken up into the cell. So here, removal of calcium, you see the cell border staining is lost, and here the molecule is actually taken up in to the cell. Now, a completely different sort of arrangement and structure is true for the other 2 kinds of junctions that I presented, the tight junctions and gap junctions. In the tight junctions, you actually have a complete fusion of the adjacent plasma membranes, and if you were to sort of rip the cells apart, you could see that these fusions are sort of little lines where the cells are connected, and you find this sort of interlacing structures of multiple lines between the cells.
These cells never break into being single cells. They always are a collective.
If a cell would be a static structure, yes.
Ah, okay.
It's not, right? Yeah. But yes. Yeah, they're attached there. And so actually what would happen, if, and this is an experiment shown here, they use an extracellular tracer. Yeah? So this is one cell, the other cell is the plasma membrane, the black part is the tracer, and the tracer doesn't permeate between the cells. Yeah? It's a little bit different for the gap junctions, where also 2 plasma membranes come in very close proximity, but there's still a tiny gap left. That's why it's called gap junction, which is in the order of 4 nanometers, a bit less than the plasma membrane itself. And the reason why I'm discussing both at the same time is that the transmembrane molecules that make up these junctions are of the same type. They are those that go through the membrane 4 times. In the case of the gap junctions, it is the so-called connexins, which actually go through the membrane 4 times. 6 of these molecules arrange to make this very nice sort of rosette type structure. And this rosette type of structure between neighboring cells connects to each other. Each of them is called a connexon, and 2 connexons, when they actually face each other, they form a transcellular channel. So in this case, we have even a direct connection between cell 1 here and cell 2 through these channels and small molecules. For example, ATP ions can pass between the 2 cells, which is responsible for the electrical and metabolic coupling of neighboring cells through these gap junctions. In case of the tight junctions, we also have these kind of transmembrane molecules, and there are two actually not groups, there's a single occludin molecule and actually a large multi-gene family encoding the claudins, which attach to each other but don't form transcellular channels, but probably, at least the claudins may have channels in the little ridges here, so they can selectively allow passage of certain molecules between the cells in this part here. All right, so we talked about the importance of transmembrane molecules, the cations, the immunoglobulin-like, example was the nectin and the tetraspanins. There are other cell adhesion molecules, particularly those that are responsible for the attachment to the extracellular matrix, the integrins, making contact to extracellular matrix components, the selectins, which bind to carbohydrates, or the mucin-like cell adhesion molecules, which also bind to extracellular matrix components and may also bind to selectins. So even this combination is possible. And this shows you just how complex the whole situation is. So we have cell-cell and cell-extracellular matrix contacts. We have homophilic contacts, and this was the original concept actually for the cadherins, you have sort of a morphogenetic coat on the surface of a cell, which would allow to sort cells of a certain kind. So cells are social, they like alikes, and this is mediated through the cadherins, which is not 100% true. We now know that even the cadherins, the classical cadherins, can also form heterophilic complexes. And then we have, and that's what I will be I'm mostly talking about the junctional components, which are located in the structures that I just showed you, but they are in an equilibrium with non-junctional molecules, which kind of shuttle back and forth, and there are probably also some that never really are part of a distinct junction. So this is the kind of complexity we are dealing with in this situation. Now let me come back to the main topic, that is to migrating cells and to what's maybe happening. And this is just some of it is very recent, in particular work from Sandrine Etienne-Manville's laboratory, which I will briefly present here. So if you have your epithelial sheet, or in this case it's actually wasn't epithelial cells, but it doesn't really matter, you have on a time lapse here on a probably minute scale, sorry it doesn't show the time here, you see that these junctions are quite stable. In this case would be the junctions that are characterized by the classical cadherins. In this case you have the desmosomes, and you see there's some sort of fluctuation, but the cell-cell adhesions are stable. Now in this wounding assay, and both of these shows here wounding assay, you have the free cell edge here, would be here or here to the right. And what you see then all of a sudden here is that these membrane molecules, or these cell-cell adhesion sites, that's what we take them for, are actually not static, but they move. So this phenomenon has been originally described by Takeishi and is referred to as the cadherin flow, which is characteristic for the leaders or for the free wound edge cells here. And from actually a large number of observations from Sandrine's laboratories, she came up with sort of a model that's summarized here, and I'd just like to go through this very briefly. So what we were seeing is actually these transmembrane proteins, the cadherins. So so we have here leader cells, and you saw that these complexes were moving down along the lateral membrane of these cells here. And this lateral or backward movement is actually driven by the actin retrograde flow. So you also have the transversal arcs here, and the whole thing moves backward. So the treadmilling of these cells that have to move forward is actually coupled to the cell-cell adhesion sites here. And she could further show in her work that this backward movement is probably driven by phosphorylation of a linker molecule in these junctions, this P120, which I briefly showed before, which through enzymatic activity becomes phosphorylated successively from here to here, which will then eventually result in a sort of probably loosening of the clustered arrangement of these cations at the back of the cell, and then the cations will be endocytosed, the back of the cell, and they're being moved forward to a microtubule dependent mechanism and going to the front of the cell. The cell probably will have translocated this way here, will be incorporated. Again, you have another cycle of this sort of roundabout movement here. Just showing you these junctions, although if you look at a tissue, are rather static under certain situations, and very rapidly they become highly dynamic and can be coupled in various ways to the cytoskeleton and to changing cell position in a given monolayer. So something like that might be happening. We had our wounding assay. Cells move forward and then they meet each other from the right part to the left part, and just reminds you, I picked this picture here from textbook again to show you what kind of processes we have to deal with. So we have we have individual cadherin molecules which will attach to each other. These forces are probably very weak. So it takes more such cadherin molecules. It's like a Velcro. So you have 1, you have 2, you have 3, you have 4. So you get sort of a small mini cluster. And this clustering will then also serve as a nucleation site for actin filaments. And this clustering and the association of actin filaments will then sort of be a self-propagating process by which the clusters become bigger, and then also the actin organization becomes more regular, for example, through RAC activity, and these clusters become bigger and bigger and bigger, and they now become highly organized in that what I've told you about are junctions that we then can see by electron microscopy or even in high resolution light microscopy using fluorescent tags, and then the actin filament system becomes organized to also additional tension that's important there into the contractile actin and myosin bundles. So cells meet, we find cluster formation within the adherence junctions, and they are formed first before desmosomes are formed. So there seems to be an ordered process, first one and then the other, and I think one of the probably most extreme examples is shown here from work from Sergei Troyanovsky's laboratory, in which he actually imaged the appearance of cation clusters and the nectin clusters. Remember, the nectin was also sort of an accessory component of the adherence junction. And what he sees here is, so these two cells approach to each other, start to kiss each other, and first you see a cluster, this is this bright spot here, of the classical cation, in this case E-cation, and just 2 seconds later, or let's say maybe 4 seconds later, you find clustering of the nectin in the same spot here. Shows you know it's a very complex and a hierarchical ordered process that takes place during cell-cell adhesion formation. And this extends also to the desmosomal junctions, which apparently need E-cation to form, and we really don't know much about how this whole process comes about, and there's some biochemical experience evidence available that's summarized in this work by the Waschke laboratory, where you have distinct complexes that have been biochemically characterized and which are believed to form sequentially. So you have biosynthesis of these membrane proteins in the cell, and these transmembrane proteins, here the desmosomal cations, are transported in a complex together with signaling molecules and with linker molecules to the cell surface. They then need to sort of interact with the classical cation, which then will serve to elicit a biochemical reaction and also activate these linker molecules, which then support clustering of these transmembrane proteins. And this process itself is incredibly complex and specialized. You may often wonder why this is necessary. It's necessary because it gives you a lot of options of differential regulation of cell-cell adhesion. That's at least my explanation. So even the different desmosomal cadherins, whether they are of the desmoglein or the desmocolin type, will actually reach the cell surface in different transport compartments. So they use different motors. So one uses the kinesin 1, the other uses the kinesin 2. Don't ask me why. And then they come to the cell surface, and only there are they brought together. Maybe I have a reason why. One reason could be that if you have a cell-cell or plug formation or these clustered arrangements, it would sort of clog up the biosynthetic machinery, the endoplasmic reticulum, various other compartments.
Is there some hierarchy of the binding strengths of these? Cadherins may be weaker than the nectin, etc., etc.?
Not that I know of, no. No. There's a fundamental difference between the classical cadherins and the desmosomal cadherins. We'll come to that, but within the desmosomal cadherins, nobody really knows, and there's actually no experimental data available at this point, no. Yeah?
This P38, is it the kinase phosphorylating keratin?
Yes, it does. Well, things do many things. There's no, yes, but I don't know what the connection is. Yes, the keratins are targets of P38 MAP kinase, but I don't know if this has anything to do with desmosomal assembly. No idea. So you have the separate transport of the transmembrane molecules, and then once they're at the surface for the desmosomes, you have the sort of, I always think this is sort of a cross-linking process that takes place. You have the linker molecules, which will sort of, while the transmembrane proteins do the trans interactions between the cells, on the cytoplasmic side, you have the linker molecules, which sort of put these transmembrane receptors together and recruit the plaque material. An important molecule that's actually guiding this process is the plakophilin in case of the desmosomes. I said desmosomes differ from other cell junctions in that they, and I really like this idea, although it's probably formally not proven, but I think it's a very good idea that's been put forward by David Garrett a number of years ago. They exist in a calcium-independent state. He calls this the hyperadhesive state. Desmosomes are present in tissues, in epithelia in particular, that are very, very resilient, and where the tight attachment, not the dynamics, are really, really important. And that's a property that's not shared by the other adherence junctions. It's unique for the desmosomes, that when you actually remove the calcium, it's not like I showed you in the previous cartoon, they become floppy. They stay the same way. And probably there's a structural difference between the desmosomal cadherins and the classical cadherins, which is also evident in electron microscopy, because these desmosomes form in the middle between, so these are these cadherins, and this is the sort of exactly the middle of the extracellular space between two desmosomal halves. You have the so-called midline, which is characteristic for desmosomes. Yet also this hyperadhesive state can be broken, must be broken, when cells become motile, become migratory, and this is actually done enzymatically. And an important enzyme there seems to be calcium-dependent protein kinase, PKC alpha, which upon wounding becomes activated, has many, many effects in the cell. One of which is actually recruitment to desmosomes, and within desmosomes, phosphorylation of desmosomal proteins. One of it is actually desmoplakin, one of the linker molecules, and this phosphorylation will then result, it probably has multiple targets, we don't know the targets, all of it yet, will then result in loss of this hyperadhesions of these transmembrane receptors, and then the desmosomal adhesion sites can be loosened and actually can be even endocytosed, and the cell can now move and is more motile. Hyperadhesion in desmosomes. Another way to see how junctions and the cytoskeleton interact and communicate with each other is shown in this kind of experiment, in which I think Reinhard briefly mentioned this. You look at cells that either actually keratinocytes that either contain keratin or do not have keratins. So there is such a thing, at least experimentally, that we have is keratin-free keratinocytes, and this is a sort of a en face view of desmosomal junctions. And you see here highly ordered structures in the presence of keratins. In the absence of keratins, you see these structures are not so ordered anymore. They are more motile. Actually, when you look in electron microscopy, the junctions themselves are much smaller. And when you take a tissue sheath, here for example, and you kind of shake it and you add some enzymes to it, you will see fragments. And this effect is actually, or in part, I should be very, very careful here, linked to release of this PKC alpha, which I just mentioned, change from hyperadhesion to not hyperadhesion, presumably because the cytoskeleton acts as a scaffold to which this PKC alpha can be sequestered. The scaffold is missing, so more PKC alpha is available in the cytoskeleton and may now sort of phosphorylate the desmosomal components, particularly desmoplakin, which will then result in reduced desmosomal adhesion. That's at least the theory here behind it. Okay, I always forget to look at time. How much time do I have?
Maybe 10,
10, 15 minutes? Yeah. When should I get done? You have to tell me.
Quarter past. You started at 3:15. So.
I started 3:15. Oh, that's why you look so exhausted. Right. I can make this really short because this part is really the journey into the unknown. We really don't know much about this. It's the epithelial extracellular matrix connections. So we have the comes again in two flavors. We have hemidesmosomes and we have focal adhesion types. Again, we have we have transmembrane receptors, which in both cases are of the integrin type. We have linkers, various components, and we have for the hemidesmosomes the keratin filament cytoskeleton, and for the focal adhesions the actin filament system. And if you look at a epithelial cell, you have at the basal cell layer, you have these half desmosomes, which look, I don't think they really look like it, but the morphologists thought they look like half a desmosome, and they are only located at the basal cell layer here in green, and in the top cell layers you have the desmosomes. Note, even though they're called hemidesmosomes, they differ completely in their molecular composition from desmosomes. So hemidesmosome, unequal desmosome, except for the anchorage of keratin filaments. Now, these hemidesmosomes are beautiful and highly ordered structures. So if you have, for example, this oblique view, they don't just, put their little feet down there, but they organize themselves in these ribbon-like structures here. And unfortunately, there are human diseases, again, autoimmune diseases, where these structures are targeted. The autoantibodies actually localize precisely here at the junction between the epithelium and the underlying connective tissue of the dermis. And as you would expect, with these antibodies result in loosening of the contact of the basal cell layer to the connective tissue. You have blister formation now here at this junction here, and this is the clinical phenotype that's associated with it. For a long time, we didn't think there was any such things like focal adhesions. I'm still not so sure they are, but there's a disease, and this disease is the Kindler syndrome, and in this case we have mutations in the kindlin gene, and kindlin is part of focal adhesions. It's clear, at least in culture, you can actually localize kindlin to focal adhesions. And in this case, these patients have these sort of skin, different appearance of the skin, you know, this funny picture there, but they have usually no blisters, but skin atrophy. They have mucosal inflammation. Only upon trauma they will develop blistering. They have photosensitivity. But if we actually look at the distribution of this kindling protein, it's not restricted to the basal cell layer, but is present on the membrane throughout the epithelial cell layer. There's something there, but it's difficult to understand. But at least we should take both into consideration. And again, it's like with the cell-cell junctions. These are molecular machines which consist of various components, and I will not go through these names again, but they're distinct between focal adhesions and the hemidesmosomes. And of course, you can either take a view, what does it take to make such a junction? This has not been rigorously done in either case, I guess. Or you can say, you know, how complex is the machinery? And this is what's been published a number of years ago, and this is certainly not complete. This is from 2007. So these machines or these molecular assemblies interact with lots and lots of components. So a term that's been coined is the adhesome for the focal adhesions, which sort of maps the interactions of the transmembrane receptors of the integrin type, and you see how complicated it is. So here they mapped 156 components, and they have multiple interactions that participate in it. That's one way to go about it. Another way, which is now possible through improved imaging by high-resolution imaging, is actually that we, and this becomes very interesting, where we actually sort of place the individual components, and if we understand where a component is, we may understand a little bit about its function, because function is very much determined by where you have a given molecule in a certain mini-environment, so to say. So this is the work by Claire Waterman. It was published in 2010, and you see here the membrane with the integrin receptors with the linker molecules. Now it gets, you probably become sort of dizzy what's in there, but it sort of makes a clear picture of what's happening there, and we need this to understand how these machines work in a normal tissue context, but also, of course, in a migrating cell. And this is the sort of last, I guess, no, there's probably a bit more, series of pictures about the complexity of the integrin receptors. I told you, focal adhesions have special integrins, hemidesmosomes have special integrins. So the focal adhesions are characterized by alpha-3, beta-1, and also other combinations. And for the hemidesmosomes is the alpha-6, beta-4 integrins. So if we think about a wounded epidermis, here again, collective cell migration, something must be happening here in the leading cell. But of course, this cartoon, at least in this simplistic way, cannot be quite right, because also these adhesions, if you have it, they must be loosened, they must be dynamic, and probably coexist in the cell here with these other adhesions. So apparently multiple options how these extracellular matrix adhesion sites become plastic. That they do is obvious. And this just shows you 3 images taken from a time-lapse series from Suhuta and colleagues, in which you see that this cell is actually translocated in this across this line here. And again, like also in the first movie that I showed you, the focal adhesions in green move from here to there. And similarly, the hemidesmosomes also have to translocate from here to here. And this is probably mediated through a crosstalk between these different junctions, the focal adhesions and hemidesmosomes. This process requires that these junctions are transient. So adhesion sites have to assemble at the leading edge and have to disassemble at the trailing edge. And the whole process must be, in one way or another, coupled to the cytoskeleton, in particular to the actin filament system and to the intermediate filament system, which in turn also determines the properties of these sites, the strength of adhesion within these sites. I think I should probably skip this here and also skip this here. We have heard about the maturation of the focal adhesion sites from the nascent adhesions, focal complex, focal adhesion, and fibrillar adhesion. Much less is known about the hemidesmosomes. There are two types of hemidesmosomes. There's one that only consists of these three components, the BP230 and the integrin membrane receptor and the plectin molecule attaching to the keratins, or these type 1 hemidesmosomes which actually have additional components. So probably there's one may be an early stage, the other one may be the more mature or stable stage, and I don't know what's happening in migrating cells. Another thing as to how to build hemidesmosomes is probably through, again, sort of a clustering process, which has been proposed very recently to be mediated by plectin dimers. This plectin is a very long, very big molecule which is able to bind to one of the integrins, actually the beta-4 integrin, and then serve such large plaque-like or clustered transmembrane receptors. And again here, same thing is true. The system not only assembles, must also disassemble. And again, I just wanted to make a point here. Protein kinase C, alpha is important, which we know from the keratin-free cells, and in this case also modification of the integrin receptor, which has been actually examined quite well in the Sonnenberg lab during the last years. And this integrin beta-4, excuse me, can then actually be released from the junction and have an alternative function in the lamellipodium. We don't have hemidesmosomes in the lamellipodium, but we do have beta-4 integrins in the lamellipodium. Actually, they may actually latch on to actin filament system, and once it becomes more mature, more hemidesmosome-like, so to say, will only then attach to the keratin filaments in these cells. I have 5 minutes more of this, but I can probably skip this. I don't know if you probably have talks about mechanosensation, right? So you will talk about this, so I should probably take this out. But it's just a reminder to you. These junctions not only transduce forces, but they can also sense forces. So they have built-in, so to say, mechanosensors, and this mechanosensation is then important to actually stabilize the interaction with the cytoskeleton, and one thing, and it's also important to elicit processes in the cells through signaling. And the basic principle as how this is accomplished is there are molecules which just by pulling on these molecules unfold, and by this unfolding you actually reveal binding sites for either molecules that bind to the cytoskeleton or other signaling molecules. And that's what we understand with mechanosensation. And yeah?
Is there only one activation site in the taline or more?
Activation site?
Sites where you
here.
coming in.
No, there's actually 5 binding sites just within these 12 helices. There are multiple binding sites. Yeah. And this is maybe I should say, this is a wonderful experiment done by the Sheets group, where they actually took out just this one fragment of this very long molecule, which is in the focal adhesion plaque. Yeah, they attached it to a surface, and then you have a magnetic bead on the other side, and they just pulled on it, and then they looked for binding of actually fluorescently labeled vinculin to this molecule. And this way they showed that the binding sites are actually uncovered. But there are multiple binding sites there already. And just to come back to our initial experiments, and this is where I will end. They actually, this work by the Spatz group, and that's where they used what I showed in the beginning, the stencil confinement of cells, of a cell group. So if you take off the stencil, then at the free cell edge, you will find formation of basically lamellipodia, or formation of a leading cell here. And they actually screened the system for this transmission of forces, the orientation of the cells, actually using this this PIV system, and identified a component that is part of the cell cortex that is associated with cell junctions. And this protein is called Merlin. Under normal situation, it prevents lamellipodium formation. Now we pull on this, the cell itself pulls on the cell behind it, and there's a force generated. And this force will then release Merlin from the junction. Actually, this is tight junction associated for the most part. So we release this molecule, and Merlin will then, so let's first look at the trailing cell, and it will now, in the follower cell, activate RAC, and RAC will actually then help to form a lamellipodium, here, see underneath here, sort of following the cell in front now, here. It's a little bit more complicated in the leading cell, because obviously the low activated RAC in the back of the cell gets activated in the front cell, will then result actually in continued binding, or again recruitment of Merlin to the junctional area and stabilization of this junction at this point. So this is one way, in one way you activate the cell to follow, but it's tightly coupled to the leading cell in this position here. And I will skip this, although it's interesting. And what I wanted to tell you is junctions are circumscribed adhesion sites. They're essential for epithelial sheet migration, as far as I can tell. They come in multiple flavors. I tried to tell you a little bit about it, and it's highly dependent on the cell type. These structures can become highly dynamic, can also be static. They cooperate with each other, not only the cell-cell junctions, cell-extracellular matrix junctions. They coordinate cytoskeletal organization in a reciprocal fashion, and they're important for force transduction and mechanosensation. And these are some of the questions that I thought might be interesting to think about. Okay, thank you very much. These are some of the people who contribute to the work in our laboratory. So we have quite a nice group. Maybe talk about this some other time. Yes?
So everybody looks at these drawings from Albert's, and you've shown some wonderful micrographs, and there's a bewildering number of different types of cell-cell junctions. Every drawing from the textbook is in 2D, right? And the plasma membrane is a little curve, and it's got these little grommets, rivets, whatever.
Well, this is the one drawing I showed you was almost 3D, because you looked in the inside of the cell, right? And you could sort of imagine that this is a 3D structure.
So my question is, two membranes up against each other, what is the shape of the cell-cell junction?
It's very different, and that's why they're called differently. So if you talk about this original initial picture that I showed you for the simple epithelial cells, we have the zonula, which actually goes all the way around the apical surface. So this is all the way, like this here. Like, yeah, if this is a cell in my head. Yeah, it goes all the way around.
A headband.
Yeah, this would be the tight junction, adherence junctions. And then we have the spot-like junctions. These are desmosomes. These are sort of circular, roundish structures, several micrometers in diameter, which are just spot wells. Cells are put together in the little puncta. And then you have the gap junctions, which are also sort of patchy areas, which are not so well defined, but are patchy areas where the cells come together. So yes, we do know about the spatial arrangement of these junctions.
The desmosomes are of a defined size, then?
Well, it depends again on the cell type, but they can be small, less than a micrometer. They can be big, I don't know, up to 5 micrometers or so in diameter.
But within a cell type, they're fixed.
As far as I know, yes. But I think people have not systematically measured that.
But then the type.
It's a good question. What determines its size? No idea.
Then
Why don't they just grow?
The tight junctions, that's a band.
Yeah.
The band of fixed width.
It's actually multiple bands, because you can. The more bands you have, so there can be actually individual lines, bands, which interlace. So the tighter a junction is, the more strands, they say, you have. Yeah, there could be one strand to, I don't know, 10, 15 strands that kind of encircle the apical part of the cell. Nature has its ways. Yeah, I don't. Yeah.
One question is, do you think that this collective migration in epithelial, or the mechanism that you have explained, could be extrapolated to other collective migration, like for example in angiogenesis?
That's what people say. Yes, the tubule formation, angiogenesis, is also a good example for collective migration. Yes.
Okay, so there another question that I have is, when, for example, in tumor you have extravasation in one tumor cell, and normally the junction between the endothelial cells are broken. You think that there the process is purely mechanical, or also there are signaling between the tumor cell with the endothelial cells to break and to break the barrier between the two cells? What do you think about this phenomenon? I think it's very difficult and it's different, but I would like to know your opinion.
There are definitely mechanisms in place by which just any kind of endothelial barrier can be broken through enzymatic activities, but there are also different ways. So I always thought, yes, you always need the junctions for invasion, yeah, and you have to open the junctions. But there's also good evidence now that some cells may actually go through the cells, transcellular migration. These guys don't care. This is relevant for immune system, for example, yeah, more than for tumors. I really don't know the literature well enough to say anything about that. Okay.
So you meant, I mean, in this great pairwise discussion of intermediate filaments, you talked about the lack of modes migrating on intermediate filaments. But is there any evidence that there is a tensional force between hemidesmosomes along the base pipette?
No. I thought you would ask something else.
well, then answer what I was going to ask, because it's probably more interesting.
I don't think there's not that many people working on hemidesmosomes, except maybe one could Sasha was reminding me of these experiments by Michel Labouesse, who's actually shown that you can initiate hemidesmosomal signaling by forces. And he's done this in C. elegans in a very elegant way. Very highly recommend to read this paper. Then he initiates a cascade of phosphorylation, which actually also affects, C. elegans also has intermediate filaments, affects intermediate filaments, which I like. There was, yeah.
As I perceived it, you showed that they firstly aggregate and then they bind to actin network at some point of your talk. So my question is actually, in general, how do they start to aggregate? Why would, for instance, these scatterings, in all your figures, they are aggregated? What is the basic mechanism underlying that?
I think initially this is just by chance. Those cells bump into each other, and probably you will find even trans interactions, which are transient because they're not strong enough, they're gone. And once you have enough guys there, let's say 5, 10, I have no idea how many you need, then it's a self-propagating process. Yeah? Then it's sort of stable enough, and then you get actin recruitment and it's stabilized and it becomes bigger and bigger and bigger, and you have the signaling events, the activation of the RAC, and so on and so forth.
Okay, but before linking to actin, do you think that only membrane elasticity can mediate that aggregation? Because if I'm bound and I pull down, and I do this on several singular points, if I'm membrane, for me, it's better to have these collected just from the perspective of membrane elasticity energy, bending energy. Do you think that this can mediate this process?
I can't really comment on this.
So,
To me, it's more like a stochastic process. If they come into vicinity, I don't know how the
they come
membrane
into
takes
vicinity.
place here.
Actually, membrane does
Yeah.
this, but if there's separated membrane does this and this and this. So for membrane, it's better that they are together, so it does this, because simply I have only one and one bending here. I just have less of bending in the system. I mean, it's quite shown that actually in Rudy's group that they have shown that this happens on a substrate. My question is just, do you have feeling that this could happen here?
I would rather pass on this question.
It happens in model systems where you can show this quite clearly. If it really is, if the effect is strong enough to affect a living cell, this is a quite exciting question. I would say we should also pass it to Anna Smith, who will give a talk next week.
Okay.
And she has
Actually,
done
tomorrow.
yeah, sorry.
Okay.
and she has done quite extensive simulations on that, and
Okay.
I think it plays a role. But you know how it is with simulations, you can always make points of discussion. So I think in the living system there is not a definite proof. Not that I know of.
All right. Thank you very much for all this discussion and input, and glad you survived. I don't know if everybody survived.